Thank you, Tim. So I'm going to ask our speakers to come up to the table for the next panel discussion, and you can choose your alter ego, since these are the names from the previous session. Oh, I see, we're getting new names. So we have a nice, healthy 45 minutes for questions. And again, I'll just remind everyone that as we get close to 12:45, we'll have the speakers and organizers go get lunch first. So as people gather for questions, I see Nadeem is already there. I'll ask one question to kick this off, because there's a theme that came up in nearly every talk. And I'll actually borrow a little quote from Lon's talk, but this was highlighted by everyone, which is that the separation of basic sciences and translation is still there, and it may be getting worse. You know, we saw the examples of when it's done well in Tim's talk, that it really accelerates things. So my question to the panel is, how do we improve this gulf? And we saw some examples. And in terms of pre-competitive investment, is it best spent on the human data side or on the tool side? Go ahead, Tim, thanks. You need to press the...
I guess that one of the issues is the extent to which our investment in academic biomedical research is directed at understanding the biological consequences of the genetic insights that we get. And maybe we have to think about a more directional approach to the way we make those investments.
So I think the risk of being a little provocative here, I think the historical model for the relationship between pharma and academia, and I've been on both sides of this one, was pharma throws money over the wall and hopes something comes back. And actually, that has worked on occasion, and it provides funding, and there's something to be said for that. I think it's getting too complicated on both sides for that to be an efficient approach anymore. And I think so that, and then it's getting more complicated on the drug discovery side, both on a regulatory environment and just on a modality, on biomarkers. And it's getting more sophisticated and complex on the basic sciences side. So I think that model has to go, personally, and we need to find different ways of funding, of bringing together the expertise of the individuals that are relevant.
Go ahead, Sohini.
I would agree with that. I think that drug development in and of itself is now so expensive and can be so lengthy that I think we have to start to put stakes in the ground about areas, and I really loved what Lon said, areas where it's pre-competitive, and maybe we need to rethink what is pre-competitive. I think big data analytics are going to change the way we think about things. I think the reality that we're going to have to go back and study humans, the Precision Medicine Initiative is a costly endeavor, but potentially a profoundly impactful one. I think we just need to start to think about what actually is competitive versus pre-competitive. And I do think that it's the investment that one has to make, whether it be in tools or in sort of clinical data sets, both can be pre-competitive. But at the end of the day, following individuals for a long time period is just more expensive. Being able to work together on tools, reagents, and assays and things like that, and being able to share it, while it may be complex from a legal arrangement, it actually is not that cost intensive from a financial perspective. So I don't think it's an either or. I think the reality is more is actually being placed under the umbrella of pre-competitive. Some groups are slower to realize that than others. But I think that's the trend if we're going to actually make progress and still maintain drug development in a realistic fashion.
Thanks.
Yeah, I do have good news from an academic perspective. The good news is there are lots of people who think that this translation is bad and that basic science is good, and they're almost without exception people who have never done it. And then all you need for them is to make a basic science discovery that they realize through some mechanism might actually have impact. And then all of a sudden I've seen entire attitudes turn around 180 degrees. And it's really just lack of familiarity and a little bit of, I wasn't trained to do that, so I'm not very excited about it. So I've seen that for colleagues, even old colleagues like me. Among young people, at least at Stanford, there is routinely people coming in saying, I'm a basic scientist, young people. I'm a student, I'm a postdoc, I'm a basic scientist, but I've gotten interested in translation. And then this even extends to people like computer scientists and engineers, where the first conversation is, let me teach you how to spell biology, and then we'll talk about how you can help revolutionize it. So that's good news. The bad news is there is a certain role for clinician scientists that is important. And if you've looked at the numbers, the NIH projections for clinician scientists look very, very bad in terms of the one statistic that I might be getting wrong, but it's roughly correct, is that there are more people above 70 who have R01s than there are below 40. Something like that. That might be wrong, so please don't tweet it yet. But there is a terrible statistic about senior scientists and junior scientists and so I think there is something we have to work on in terms of the educational pipeline. But I do think there is great interest there, and so I'm optimistic that that we can have the kinds of alliances that Lon was just referring to, which is not throwing money over the wall, but saying, we have some interesting data here at this drug company, we have some interesting problems, you have some interesting capabilities, let's develop a relationship and have an actual collaboration. And I'm actually bullish on that model.
That's great. Sally.
And so I think, Tim, you ended your talk saying we need to understand what DNA variants are doing at the cellular, tissue, and whole organism level. And that does require enormous investment in tools and reagents to understand biology of areas that we don't really understand, like cholesterol metabolism, for example. And I think that is largely pre-competitive. I also think one of the things that for me stands out about the genomics community is the collaboration and also the robust and reproducible nature of what we publish. And I think this morning we heard about biomarkers, thousands and thousands of published biomarker studies and about 100 validated biomarkers, and it is less sexy, but if we can do that pre-competitively and come up with more translatable, robust biomarkers that we can use in the clinic, that would accelerate our ability to translate the basic genomics.
Thanks. Nadeem.
Thanks. I'm going to exploit my power as chair and ask two questions. The first maybe for Lon, and the second maybe for Russ and Tim. So Lon, you provided a very nice graphic. So, well, first of all, and we're a self-selected bunch, it seems unimpeachable that human genetics is informative for drug discovery and increases your odds of success. Kind of flipping that round, it almost seems unethical, therefore, to pursue less validated therapeutic targets. Your slide suggested that's what we do. So over 80% of therapeutic targets would not be validated using human genetics, using our criteria. Why is that?
I don't think it's a great question. We're asking it too, and I don't think there's a single answer. The first one that would come to mind is we had these findings only for about 10 years, and it is a long road. So some of this just takes a while for culture change and for uptake and so on. And those findings that I was describing are in the clinic. So they went through their preclinical phase and their chemistry and their years of all that. So I would like to think we're just seeing the tip of that, and they should start coming through. That's part of it. I also think we should be realistic about the fact that I've forgotten who raised the point. Genetics has had its ups and downs, and there was a lot of hyperbole around the genome era, and pharma bought in a significant way, did not see immediate returns, and jumped out. It's the proverbial classic hype cycle, and it happened in industry. And so those things take a little while to come back up. And I do believe the field has come back up and continues to do so. But that and just the time have conspired against us.
Sally, you wanted to add?
I was just going to comment on the types of genes that we've found a lot of, which from case control studies are susceptibility genes. So we found a lot of genes that affect the risk of getting a disease. And in the non-genetics community, there's some degree of skepticism that genes which increase susceptibility to disease are going to influence and be disease modifying once you have disease. And I think the world of cardiovascular suggests that it is the case
But if you look at cancer, it's if you stop smoking, once you've got lung cancer, it's not the case. And I think we don't necessarily know where all of our diseases fit on that trajectory. And it comes back to, if we understood the biology better, we'd have a better hypothesis.
Actually, an idea in real time, to your point, Lon, maybe some of the people in this room could get together and report what proportion of our preclinical pipeline is genetically validated, and is that a significant change from 10 years ago? I wonder if a group of us could get together and have that chat. My second question, maybe for Russ and Tim specifically, or the broader group as well, I was pretty struck by the consistency in message. So the key bottleneck or enabler that you all identified was better understanding of the biology. And Tim, I really liked your phrase at a cellular, molecular, and whole body physiology level. And so you highlighted very nicely the importance of biomarkers. You all had it there, but no one put at the top of your list druggability. And I think there's often this conception that in fact is the key decision maker. And I was wondering if maybe Russ and Tim, you could comment on that. Is druggability the key enabler, or is it understanding the biology?
So actually, perhaps we should put up the slide about NAV 1.7, because if we can.
So
Or
I think it was
it's got
Tim's
the
last slide.
four targets on, because I'm going to try and answer your question on that. So, NAV 1.7 is pretty, or SNC9, SCN9A, I think is potentially pretty informative to us at a number of levels. So just recently, John Wood, who you can regard as the father of the biology of NAV 1.7, published a paper which phenotyped in I guess in an unbiased way, the transcriptome of the dorsal root ganglion of mice have had NAV 1.7 knocked out, and as controls he did 1.8 and 1.9. And what he found was a very marked upregulation of proenkephalin. And he did some very nice experiments that showed that naloxone reversed their insensitivity to pain. That's the mu-opioid antagonist. So clearly, loss of function of NAV 1.7 leads to a chronic adaptive change and he went on to provide the first evidence that that's determined by the levels of sodium within the neuron. And that's why I made a comment earlier about, sodium channels gate, they leak sodium all the time, and it's the pumps that keep the sodium level down. So maybe what we should have been looking for was a pore blocker, not something that stabilized it in the activated state. But I think if we were looking for a pore blocker, we would have known that probably was never going to be druggable from a selectivity standpoint. But I use that as an example, was that the person 10 years ago who was the author of that loss of function paper is the person 10 years later who comes up with that data. And you can look at MC4 and say the same thing. We got peptides that suppressed appetite, but they increased blood pressure. We then went and looked at the clinical phenotype, and lo and behold, people who have loss of function of MC4 have, I'll get this right, have lower blood pressure. And so we kind of equated that. We couldn't separate the satiety, the induction of satiety, from the induction of sympathetic tone. But recently, with the application of novel neuronal pathway elucidation technologies, it looks like there are separate neurons within the hypothalamus that are mediating the sympathetic tone from those with satiety. And not only that, the GPCRs in those neurons, the MC4R, has different downstream trafficking through G proteins. So that actually gives us a reasonable basis now to mount a program based on a sort of rational approach. But look, when 1998, 18 years later, we get what could turn out to be really pivotal information. And that's why I keep sort of going back to the biology. We made a mistake with NAV 1.7, and that was hope springs eternal. The mouse knockout has no response to pain. We put our compounds in mice and we get a kind of ho-hum response, okay? And we took that to man, and what did we find? A ho-hum response. We make mistakes because of that hope springs eternal. But if we held ourselves to the fire on that very simple track, I could guarantee success every time.
Thanks. So, yeah. You want to grab a microphone? Yeah, yeah, yeah. I think, that's okay. Thank you for commenting. I think if we can distribute a microphone if necessary. Russ, did you want to add to that?
I was going to say that in the setting of a great target where you want to get that target with low nanomolar affinity, then druggability is going to be key, and there's work to be done there, understanding biology. The enthusiasm that I have for pathways is that I think it's a lot easier to have multiple shots at a pathway, and it may be that 3 micromolar binders, if placed cleverly, could give you a knockdown of some pathway that you don't like. And in fact, if you look at the trends as a physician, all the major trends for actual treatment in the clinic are polypharmacy. And so, we treat HIV with 3 drugs, we treat TB with 4 drugs, we treat depression with 2 to 4. I mean, everything where we're making any progress. And so I think that's another very challenging, again, I recognize very much as an academic, this is a very long time scale thing, but I think it's inevitable that we have to think about what I think of as constructive polypharmacy. And one of the reasons is the targetability of a pathway just statistically is better than the targetability of a single element of the pathway.
Great. I think we should move on to the other side of the room. Thank you, Nadim.
Well, thank you for such fabulous talks. I mean, there was some important messages for me. I mean, I think Tim beautifully illustrated what the pharmaceutical industry can do. When they have a target, they can translate it into a drug. Sahini nicely said what she's been doing around building new biomarkers or coming up with sort of stratified patient cohorts that we could do clinical studies in. Lon made, I think, an incredibly important point that he believes that targets are pre-competitive. Now, let me just share with you, some of you know that I used to work in industry and I've moved into academia. I absolutely believe that many, many things are much easier to do in an academic setting. It's much easier to access clinicians. It's much easier to access patient groups. It's much easier to work with patients and patient material. So what I hope together we can do maybe in the future is that academics, not-for-profits, government, charitable funders, I think our job should be to come up with new targets, new biomarkers, new ways of stratifying patients. And then big pharma industry can take that knowledge and translate it into new medicines for patients. So some gleanings from the speakers and maybe a suggestion of what I would like to see happen in the future. I don't know if there's any sort of comments to that.
Any of our pharma? No, I think it's a point. Your point is very well taken. I think from the pharma side, we'd all agree completely that would be a good development.
But can I just put Lon on the spot? I mean, Lon's a visionary, and he said target should be pre-competitive. I know in academia, many of my colleagues don't believe that. And I suspect many people in industry also don't believe it.
Yeah, I even tried to flag that. That's absolutely the case in industry. There's a spectrum, but I think we should not distinguish. We should not confuse pre-competitiveness with the fact that we can do elegant science as well. So some people will say, I think targets are that I can do very clever experiments on targets. That's fine. That doesn't mean they're not pre-competitive. It means you can do very clever experiments. If you want to share that information, great. If you choose not to share that information, that's perfectly fine too. But the targets themselves remain open to everyone. So I think I would like to think we're moving toward an era where that will be the case. None of us have a legal background here, so I think we've all stayed away from that. But there does seem to be a trajectory that's in favor of this pre-competitiveness as well. So I think that part, it's where it gets complicated to me is when you start bringing molecules into it. Where do you draw that line and how do you distinguish that? Because molecules are also tools that can interrogate the targets themselves for validation. So I think that's where the debate is to be had more personally.
I also think that, we can be creative again. We actually spearheaded an initiative with 3 Pharma who are all targeting LRRK2. It's the target. But there are some really large challenges that all of them are facing. And so they actually all shared their tool compounds with the foundation to determine whether a safety profile that was identified in one compound is actually relevant to all compounds that are targeting the LRRK2 kinase. I don't think anybody here would expect that 3 pharma companies would give their compounds to a centralized sort of the foundation, all share the data and then share that safety readout data with the rest of the community, knowing that the rest of the community includes individuals that are also targeting LRRK2. So I think that's the nuance here, is that pre-competitiveness, I think, is the new model. And it's, again, trying to define what really is going to benefit the group as a whole, because frankly, all of them benefited from now knowing this safety initiative data, versus what actually truly is proprietary. And I kind of put both pharma and academia in there. I think academia has its own issues with the Gatorade hope that everything that they develop will reap the benefits of Gatorade. And the reality is very little will become the next Gatorade. So I think this is a field-wide challenge to understand that pre-competitiveness actually can be done in very creative ways while still safeguarding proprietary interests.
Thank you. Arthur.
Yeah, hi, Arthur Holden, and I'm fortunate enough to have a little bit of experience too in pre-competitive types of organizations. And I'd like to pick up on one of the things in the first and the second session, and we've seen some wonderful examples from the foundation, from pharma coming together, academic initiatives going into countries. The theme of each of these is, and it goes back to this question of what are the disruptive technologies that can really improve our productivity. One of them is to continue to experiment and innovate in collaborative structures. Because one thing you see across all these initiatives is that where there's innovation and risk-taking and focus in a pre-competitive, you see many times big jumps in productivity. And Lon, I'd be interested in your comment on it. You showed that obviously the science and the associated informatic challenges of phenotyping over time across ethnicities in a global context, it's very, very complicated. But if you look at your slide on pharmacogenetics, and you look at the number of significant associations comparing efficacy pharmacogenetics and safety pharmacogenetics, you're struck and say, why are there so many more safety pharmacogenetic associations? When in fact, it should be relatively easy to do efficacy pharmacogenetic studies. The reason is there was an innovative pre-competitive collaboration that many in this room have been involved in that actually brought phenotyping to a certain quality and then aggregated cohorts across countries and did the genetics collaboratively and put that out so that we better understood those risks. And I think as we think about these greater challenges as we're moving on, we got to continue to encourage innovation in those types of collaborations. It's really more a comment, and feel free to add to it.
Mark, were you, okay. Carrie?
Lon, I'm struggling with this idea of the pre-competitive target, because the target is not just a molecule. The target is a molecule in a context. It's in the context of biology. It's in the context of disease. And basically, if you think about it, the most challenging aspect, Tim said before, give me a target, basically, and I can give you a drug in the end, all right? So I think basically the big discovery, and let's hope that those who are going to win the next round in the industry are the ones who are going to do it on the basis of the best science. So the science is always going to be focused on the discovery and the characterization of the target in the context that you want. So I think it would be devastating if we would make the targets pre-competitive. That's where we should be competing, all right? That's where it's going to be exciting. That's where we're going to throw elbows, all right?
I don't understand the. Firstly, let me say I'm really pleased personally to be back in the field of genetics. I left it for a little bit, and I really miss sparring with Kari. It's been a number of years, so I don't understand the distinction, Kari. So I really don't. So what you publish is pre-competitive at some point. So why are you arguing differently?
Now, I'm saying it becomes like all of this. It comes into the public domain in the end, all right?
Yeah.
And basically, we publish everything we do.
Yeah.
All right. But it becomes, there is this little time between the time when you discover it and until it's published, when it is a privileged information, all right, for the owner of Decode, all right? And to turn this into a totally sort of pre-competitive, what of this is competitive? Do you want the industry just to survive on being a sales organization?
No, actually, I want to improve the production of medicines.
Yeah.
Quite opposite of
through
where you're going.
science where people compete.
So if you don't want to share it, you don't share it. Right? There's no obligation to share anything you work on. But the information that we have, we're tripping over each other, making the same mistakes, doing the same experiments, reproducing failure, not knowing where to look in the pathways. I think it's more efficient from a societal and from a patient perspective to think of that part of the game as something we could share.
So I guess a historical perspective would be gene patents, 20 years ago, as the reference genome and ESTs were being done. Everybody rushed to file patents and eventually realized it wasn't really worth it. And so I guess your argument is we're coming to that same juncture with the current generation of genetics, and I think Sally also has a comment.
I profoundly disagree with that, because this is basically where the target you find, which is basically the key to the biochemical pathway that is perturbed in the disease, all right? That is currently where most of the innovation comes in. That's where the good science comes in, all right? And I think that to classify that as a pre-competitive, I think it's misleading. That's where the competition takes place. In the end, all of this is going to come out into the public domain. When you publish it, when you file your patents, when you market your drugs. But I think that exactly in the target and the associated biology and the associated biochemical pathways, that's where the magic takes place.
That's your entire perspective. I am, to be clear, I'm not calling for a mandate on target release of information. If you choose to pursue that, Kari, I think it's perfectly fine for you. I think you do express a very narrow vision on thinking all innovation is around basic biology. I think there is innovation that occurs at various steps along the way that maybe you're just unaware of.
Thank you. That was a really good question. Sally, I think you had some more follow-up.
I was just going to articulate a bit further the industry partnership for human collaboration we had with the Finnish biobank. So each company submitted a number of genes to the academics. So we didn't know which other company had submitted the genes, but we all saw all of the genes. We didn't say what indications we were interested in pursuing those targets for, and we have all seen all of the data that's come back for all of the genes. And I think, at least speaking for Biogen, we're very happy with that because we have seen all of the phenotype associations with all of the genes that we're interested in, and actually we are very unlikely to start a new program in something that we're not already interested in.
Thank you. Sharon.
Sharon Terry, co-chair of the Roundtable, Genomics Roundtable, and president and CEO of Genetic Alliance. So, Lon, I love where you're going, and I actually would call for a mandate for moving the pre-competitive line in the way that you're indicating. We had a workshop and then a meeting report 6 years ago, toying with the edges of this kind of stuff. I know several people, Chaz and others, were part of that. I think we need to really reflect on why we're here, which is what we're doing is not working so well, for at least from the point of view of patients, that we're not accelerating this field as fast as we could. And we know at that point we looked at the semiconductor industry, astronomy, entomology. We looked at other disciplines that are accelerating faster in terms of discovery because they're not hoarding what they think they need to hoard. And we need to think about this as an information science. We have an abundance of it. There are plenty of targets if we look at the 10,000 diseases that affect humans. So I think if we could think about how together, and to the last panel too, more than just sharing in selective ways, to share in much broader ways. The people who own this information to begin with, the phenotypes on the scales, want to share it and need to share it. And we're also not asking that every person on the planet share, but there's enough sharing in the human phenotypic realm, and there should be enough risk-taking in the pharmaceutical realm, especially since the business models of old are not working, and who's happy with 18 years, $1 billion, and a 95% failure rate?
Thank you. Does anyone have any comments? Mark?
I'm wondering if your definition of target and causes are not exactly the same in some sense, because I think there's a sense in what many of us feel, and where some of this discussion is gravitating towards, is that there's a responsibility that genotype phenotype correlations that come from our patient's DNA and the medical record of our patients are not resources that can be hoarded, firewalled, and we have a responsibility to get the most out of those resources by sharing them. And yet at the same time, obviously, there's the majority of the innovation downstream, and the biological insights that come from those are not necessarily going to be developed by the same people performing those genotype phenotype correlations. And so how to set that boundary in a way that maximizes the patient resources being used and available to everyone, but retains the motivation that's needed for the industry contribution, I think is worth fleshing out further. I don't profess to have a clear answer as to where that boundary lies, but I think maybe depending on your definition of target, you come down perhaps with a slightly different viewpoint.
Thanks, Mark. The other side of the room?
Just a comment about the patenting of targets. I think it is a very slippery road to go down to. I mean, take the example, for example, of the tyrosine kinase inhibitors. You have about 450 of them, of which about 50, 75 perhaps I've seen in a research. If you allow the wholesale patent of that estate, you basically preclude innovation with those yet to be research targets for many years to come until those patent expires. I mean, we've got companies in this room who have fought patent battles on PDE5 inhibitors, for example, where one company tried to patent the target, and Lilly, for that matter, had to spend many millions of dollars in reversing those patents so that innovation could happen around that target. And you had the same case around NF-kappa beta, where I think Lilly was taken to task again for a drug that validated a patent. They didn't exist, from what I recall the case, when the drug was approved. I mean, so you get into those really weird things. To avoid those situations and allow innovation around a target, I think we've got to perhaps take a strong stand that target is really about knowledge. And knowledge, according to my understanding of patent law, which I think is not deviant, shouldn't be patentable. It's not reduced to practice. It is knowledge. It is what you do with a target, which is, I think, the point that Lon made earlier. What you do with a target, your ability to come up with a molecule to modify, to modulate that target, that is patentable. But I think we should need to draw a line there. I mean, if we allow the patent of knowledge, if we allow to patent knowledge, we basically preclude innovation until those patent expires, and I don't think it's what we're looking for.
So I want to keep this discussion going. Bob, do you want to continue on that, or shall I let you continue, and then Carrie, please join in?
Yeah, it's all same
all
discussion. I think it's fascinating to me that the first panel and the second panel are dividing in an interesting space here. For me, I was at the Precision Medicine Summit at the White House 2 weeks ago, and Obama was very articulate, extremely clear that patient information really fundamentally belongs to the patient. I'm concerned that there is a historical precedent here that consent belongs to the person doing the consent, and if that's pharma, they own the consent. And I don't think that's the direction things are going. And I really applaud the panel here for being forward-looking and recognizing that the patients are involved in the study. The DNA sequence that evolves from the patient consent belongs to the universe of information, not to the pharma company who pays for it. And that, for me, defines a pre-competitive space. So there is something, it's reflected in Myriad Genetics, it's reflected in the PMI initiative, and I think it's reflected in some of the forward-looking pharma comments we're hearing today, that there is discovery to be made. There is a way to empower the pharmaceutical industry without stripping the individual of their participation here.
Thank you. Carrie, I think
I want to emphasize that I was not advocating that people would patent targets. I think that would be travesty. And of course, you don't patent knowledge. And I think that what we are basically doing here is debating a little bit the definition of what is pre-competitive. All right, because there is no question, in the case of Amgen, for example, and Decode, that all of our discoveries are published. And there has never been an attempt to delay publication of any of our discoveries. So that isn't the issue. The issue is just where do you look at, where do you see your advantage when it comes to discovering a drug and bringing a drug to market? All right, where does the magic take place? Where do you establish that you are better than the man next to you? Because the competition is always very important from the very beginning. And I think that what is going to draw a line of distinction between the companies that are going to succeed and the ones that are going to fail is the selection of the target and the ability to put the target in the context of human biology, be it normal or pathobiology. So perhaps Lorne and I just agree completely.
Yes, I think we may be in violent agreement here, but I think, Tim, you wanted to comment.
I think we may be in a violent agreement.
Would be the first time ever. No, I think there are some semantics to this, Kari, that are worth getting through, and maybe that's a reasonable outcome from this meeting of really drawing out what we mean by pre-competitive and where are the points of agreement and disagreement, and those that be resolved. I'd like to come back, since I've got the mic open. It is really important that we get that straight in the context of big biobanks, if you will, like PMI. Because we're talking about target validation as the earliest step, and we're thinking about basic biology and everything. But actually, we need to know what we can interrogate and who has access to that when we have things like PMI there together. Because asking questions about the specific target can be greatly enhanced by having access to data sets like the type we're about to develop. And my contention would be all parties need access to that. It can be driven, I like the patient focus. It's one of the most attractive features of the PMI, I think. It's really driven by the patient, but not just the patients, but whatever the commercial or scientific background. If that information, if we could find a way to make that information accessible, that helps the earliest stages of drug discovery and target validation, not just the later ones of pharmacogenetics and applying existing medicines to stratified samples.
So, Tim, did you want to comment on this as well?
Yeah, I guess this issue about pre-competitiveness is sort of thrown into sharp relief by the way in which medicines are reimbursed now. So, the entrance of a new mechanistic class, it's very difficult unless you've got you're prepared to invest very heavily in expanding a label quickly to enter after first or second. And in fact, increasingly you see that first entrant sets the expectations of the cost effectiveness of the medicine. And so there's a real tension around this speed component. If we all had access, all pharma companies and biopharma and so on had access to the same targets at the same time, then my guess is that some of us would go faster, perhaps if it was biotherapeutic than others, and likewise with small molecules, but it's not going to be much of a difference. So inevitably, I think the generation of insights that lead you to a therapeutic hypothesis to some degree is going to remain pre-competitive. I don't think we're going to be patenting it, But I would argue that your model for gaining genetic insights in Iceland was sort of an experiment which has been very successful, but Aris described another one using, a US commercial healthcare system, and Richard described another one with 23andMe. They're all different experiments to try and get to the same thing. But essentially, I think it's the possibility that you could get a hypothesis and get towards a drug faster than anybody else that's driving you to that. So I think we have to recognize that there is a bit of a winner-takes-all environment operating in market access. The opportunities for us to sort of refine and get a second and third generations in a mechanistic class have diminished very, very substantially.
So with my chair of the workshop hat on, I just wanted to re-steer the conversation, but this is a perfect segue to the third session, which actually is going to focus on entrepreneurial and alternative collaborative business models. And maybe in the last 5 minutes of this session, I wanted to pick up on a really important point that Sahini made. So at our company, AZI, we have a corporate objective called human healthcare, which means that all 9,000 or so AZI employees must spend at least 1 or 2 weeks a year with patients, and it's your choice how you do that. And the insights you get from that are pretty interesting. So one specific anecdote of mine, I met with and interacted with study participants, and we were discussing what's it like being a study participant. And I'll never forget, we had a conversation with a gentleman who didn't really fully understand this concept of consent and approvals and when you can or can't use data. And we were expressing to him that sometimes data collected in a study cannot be used because we don't have the consent to use it. And he got really angry, and he said, so let me get this straight. I took the day off work, I went to this place, I gave you bits of my body, you took pictures inside my brain, and you're telling me you might not be able to use it because someone somewhere didn't write the right word in a consent form. He was really angry, and it opened my eyes to this point you made about kind of the obligation to study participants to make sure that the data collected is used as maximally as possible. And I was wondering if you'd comment a little bit more about the really important point you made of ensuring kind of patients' interests are met by large-scale bioresources.
Sure. I mean, it's interesting to hear the discussion because, again, I would argue that one of the things through the debate here today is no one's really talking about the fact you have another stakeholder in the room that can become extremely vocal and very active. And technology is just going to help them become more active because even small groups of patients who may have a more rare disease now can actually connect. So the landscape of research is changing, and I think that integrating and understanding the role that patients can play is important. I think those that do not understand that landscape is changing and patients are going to get more active and they are going to question why they're spending time or why should they spend time giving information or giving resources that then cannot be widely disseminated they're going to have challenges. They're going to have to answer some hard questions because, frankly, the foundation is realistic. Drug development, at the end of the day, is a profit-making enterprise, and we have to make sure that there are incentives for groups, pharma, biotechs, academia, others who play a role in this to get the remuneration, whatever that may be, through tenure, papers, or profits, that they get that so they stay invested. We're not naive that way, but I do think the stake of what you get from patients and trying to create proprietary aspects to it has to change, and it will change because patients are going to question. The more you're going to need from patients or from populations in terms of information, the more they're going to say, well, hey, what are you doing with it and who's going to see it and why aren't you letting these other groups, because I heard this group is now working on my disease and I heard that group's working on something that I care about. Why aren't you sharing it with them? And you're going to have to find an answer, and saying, well, it's kind of proprietary or, patent or this or that, that's not going to be good enough and you're probably going to lose out on that community. So, I mean, again, I'm being very facetious and very high level here, but the point being that technology is able to mobilize, empower, and drive patient power in research in ways that I think we still don't understand. And I think the announcement earlier this week from Apple about their research kit and the number of patients who have gone online to give information, if you've gone on and you've seen how easy the consent process is and the fact that you can share data with anyone, That's attractive, and that's the new trend. So you can get on board with that or you cannot, but I think you have to understand that that is a trend that is probably going to continue and will expand.
Thanks. We're going to run out of time shortly, so I invite, short questions or comments, but
So, I mean, I think Sharon nicely said, and Nahid also said at the start, this process is too costly, it's too slow, and it's too risky. A number of our panel members have said complexity is going up. We've got tons of data, we can't cope with it, and the choice is going up. We've got potentially 20,000 proteins to choose from, etc. What this table illustrates is that even when we know a target linked to a disease, it can take us a couple of decades to translate that into a drug. I'll just leave you with the comment, ApoE, we found out in Alzheimer's, I don't know how many years ago, Lon? Close to 20? Well, it's
20.
the best genetically validated target in Alzheimer's. We still don't have a drug for a condition that potentially, if we don't come up with a new treatment in the next 15 years, that one disease is going to cripple many societies.
thank you for that comment. Arthur, one
just a
last-
quick comment. I think going back to this, what's one thing that's going to be disruptive here? And it goes back to your comments in terms of the patient, is as technology, the answer is in technology, is what's really going to change this fundamentally, and it's evolving quickly. But when you can put an individual's personal medical record under their control, their genomic sequence, again, through a third-party data provider that allows them to use that in any health situation, demands that the provider who they're paying for the service puts that data back in, and then you aggregate that with the right informatics company that then exploits opportunities to use that for research and for the increased quality of human care, much like you're getting in some of the European models where they're trying to get, it's country-based, they've got a health record, they're trying to drive the research centrally. You can't do that in a pluralistic system like the US. So I think information technology that brings, and it's going to happen, I think that's going to really revolutionize the way we aggregate cohorts and how we get longitudinal information. It's just not going to happen overnight.
Thank you. Well, again, thanks to all of our speakers for a great session. And speakers and organizers first for lunch.